Every second, your body performs a high-stakes balancing act. From the moment you stand up out of bed to the peak of a sprint, your mean arterial pressure must stay within a narrow range to ensure your brain is perfused without bursting a vessel. Today, we are diving deep into the integrated physiology of blood pressure regulation. We'll break this down into 3 phases: the immediate neural reflex, the intermediate hormonal shifts, and the long-term renal control. By the end of this video, you'll understand how the body transitions from a millisecond baroreceptor fire to a multi-day shift in sodium excretion. Short-term regulation is all about the nervous system. Think of it as the quick response team. It relies on baroreceptors, mechanoreceptors sensitive to stretch. The carotid sinus, located at the bifurcation of the common carotid, monitors pressure going to the brain via the glossopharyngeal nerve. Meanwhile, the aortic arch monitors systemic output via the vagus nerve. When pressure drops, stretch decreases, firing rates slow down, and the NTS in the medulla triggers a massive sympathetic outflow. This increases heart rate, contractility, and causes systemic vasoconstriction to pull that pressure back up within seconds. When the pressure drop persists for minutes to hours, the kidneys take the lead through the Renin-Angiotensin-Aldosterone system, or RAAS. Juxtaglomerular cells sense low perfusion and release renin. Renin meets angiotensinogen from the liver to create angiotensin I, which is converted in the lungs by ACE into the potent vasoconstrictor, angiotensin II. Angiotensin II is a multitasking molecule. It constricts arterioles to raise peripheral resistance, stimulates the thirst center in the hypothalamus, and triggers the adrenal cortex to release aldosterone. It's a bridge between immediate vascular tone and long-term fluid balance. Long-term regulation is ultimately about volume. You cannot have sustained blood pressure changes without changing total body water. Aldosterone acts on the principal cells of the collecting duct to reabsorb sodium, with water following osmotically. Simultaneously, antidiuretic hormone, ADH, inserts aquaporins to pull back pure water. But what if the pressure is too high? The heart's atria sense the stretch and release atrial natriuretic peptide, ANP. ANP is the anti-aldosterone. It promotes sodium excretion and vasodilation, acting as the body's natural safety valve against volume overload. To summarize, your baroreceptors handle the second-to-second changes, RAAS manages the hour-to-hour fluctuations, and your kidneys dictate the long-term set point through volume control. Understanding this hierarchy is the key to mastering antihypertensive pharmacology and shock management. If you can trace the path from a stretch receptor to a sodium channel, you've mastered blood pressure physiology. I hope this breakdown helps you visualize the complex machinery keeping us in homeostasis. If you found this walkthrough valuable, consider subscribing for more high-yield medical lectures. Stay curious, study deeply, and I'll see you in the next lesson.